So we will do telomeres and senescence, aging, right? Deterioration with aging, what happens as we atrophy and become our sort of hunched-shouldered old selves. And then a theme of all of this is really exercise genomics, but we'll talk more about that as we move forward. We'll keep talking about that. Really today, what we're hitting though is the telomeres and senescence, and what is the biological explanation for aging, and can we intervene, can we interact, can we halt it? Our team scores are updated since last time, and you know where to find stuff. Just keep this in mind, because at the end of today, I'm going to talk about older adults and exercise very briefly. What I'm going to do is reopen this book and go to chapter 6. This is chapter 2. All this stuff is in chapter 2. Open up to chapter 6, which is healthy people, healthy adults, and there's a page in there that talks about exercise for older adults and what do we prescribe. So we will do a very short job of exercise for older adults, and then we'll move more into the detail there. But this stuff is the foundation of all of that. You know what metabolism is. It's all of these chemical reactions in the body. We'll talk about the nutritional part of metabolism at some point during this block, only from the perspective of glycolysis and pH changes and what causes the acidity during exercise. We'll talk about individual steps in here, not every step, but particular steps that correspond to hydrogen ion accumulation or absorbing. So we'll talk a little more about that. Just remember cell signaling, right? Growth, cell signaling, protein translation, protein degradation, the balance of those two, protein turnover. Are we in hypertrophy mode or atrophy mode? Just remember that stuff. And Gregor Mendel, father of genetics, how it works, and looking at some of the mathematics of this. Not necessarily that he got the mathematics right, at least according to Ronald Fisher, but we have known for a really long time about inheritance, right? Whether it is with plants, thousands of years with plants, thousands of years with animal husbandry, right? Thousands of years of selective breeding of animals to elicit particular phenotypic traits. We want a phenotype. We know we have to breed those two that express that phenotype. And nobody knew anything about the genotype at the time or how that came together. So the rules of inheritance this is a relatively new field, and it gives us something to say about pharmacology, right? It gives us something to say about nutrition and exercise and how our genes behave and exercise genomics. It gives us a little bit more to say that's not just, okay, two tall people make a tall person. Right? Two fast horses make a fast horse. It gives us a little bit more interaction, but the genes themselves are not destiny. It's a predisposition. And everything we're doing is changing our genetic expression. The behavior of our genes changes with everything we're doing. But remember, there is everyone in this room has your own genome, and that genome is, like JP and Ryan, they don't have the same genome. And so they go exercise and the body responds differently. Everyone in the room is going to respond differently to exercise, which illustrates the importance of doing fitness testing, of doing some sort of blood work and exercise tests and check blood pressure. And because some people exercise and triglycerides or insulin or HDLs or blood pressure gets worse. Other people get way better. Some people are non-responders. And if you just give every single person the same exercise prescription, the same dietary prescription, you're failing as a practitioner, because there's a high percentage of people who are either not responding to what you're giving them or responding badly. So you have to know how to test. You have to know how to interpret those tests, and you have to understand the basic science to respond to those tests in a way that's best for your client or patient or whoever. Proteins do a ton of stuff. Now, you get, in the last 70 years, And we're starting to figure out the structure of this stuff, the structure of DNA, which gives us a little bit more input about how that structure facilitates its actions. Just, the basic vocabulary, right? The genome, that's you. That's your entire, every gene you have, the 23,000 whatever. Protein coding genes in you. And then the chromosomes, that's where all those genes live. You have 23 of those, 23 pairs. And then the genes would be the chapters. And you have about 3.2 billion base pairs, right? The A's and the T's, the C's and the G's, the T's and the A's, the G's and the C's. This is a base pair. That is a base pair. That is a base pair, right? And so you have a ton of those things throughout your genome, throughout your chromosomes. And depending on where you are in the body, you are expressing certain genes. You are making certain proteins. Remember, your pancreas isn't hairy, and you're not just squirting insulin out of your skin, right? You're going to express particular proteins in particular spots, but there's also going to be proteins you just don't express. There's going to be genes, they're just dormant. They're not doing anything. But, 23,000 or so, who cares really how many, the number, 23 pairs of chromosomes, but 99% of the genome is not actually coding for proteins. It doesn't mean it's pointless, it just means it's not coding for proteins. And we're a human being, all of us, we're somewhere between these in terms of the number of protein coding genes we have. And it doesn't really matter the number of genes, right? It matters what those proteins are and what those proteins are doing, how those proteins are interacting. And alternative splicing, remember, one gene can generate variant forms of a protein. One gene can spit out multiple forms of an individual protein. And then you can have post-translational modifications where you take a protein that has been synthesized, that has been translated, and you modify it so it takes on new characteristics, new behaviors. So, and then again, atavism, there's stuff we just don't express. But when you look at the proteasome, when you look at all the proteins inside of us, oh, there's a ton. Yeah, this is not it. This is not the answer. This is a ridiculously low estimation. We start to get into some huge numbers in terms of the number of uniquely behaving proteins inside of us. And those proteins are doing everything. You know how you come together, you're half mom, half dad, A, C, G, and T. Those are our base pairs, and you're a cell, right? There's a U for if you're mRNA. But so we have our DNA, this is the nucleus, transcription, we create the mRNA, that's the recipe or the architectural plans, and then you take it to the kitchen or the architect, and that's where you assemble the protein. And that protein folds according to the order of amino acids. Swap out one amino acid and it folds differently. Swap out a different amino acid, it folds differently. The protein will fold according to the sequence of amino acids. And one amino acid different could mean, it's the physiological equivalent of night and day, the way that it's going to fold. So you know how transcription translation work. And the ribosome, remember the ribosome is not in the nucleus. This is out of the nucleus. If it's architectural plans, the architect is drawn up the architectural plans, in the office, and makes a copy of those plans to send them to the construction crew, right, to the building site. You don't send the actual original copy, assuming that it's never going to be lost at the construction site. You make a copy and send that out to the construction site. And so that's what we do. There's also transfer RNA, which brings the amino acids here. This is the messenger RNA, right? This is what is red at the ribosome, red down here, and then the large subunit. That's what links together those amino acids to create that string of amino acids. So your genotype codes for your phenotype, and your phenotype codes for your extended phenotype, right? We have traits And, again, the bees make the hive, the beavers make the dams. Each of these, critters and creatures and monsters and whatever, they make their own specific thing. Now, again, you do anything and you're going to change genetic expression. Go eat, go don't eat, go exercise, go don't exercise, and you're changing your genetic expression. And the codons, remember these nucleotide triplets, 3 in a row, these codons code for whatever particular amino acid in that chain. And so your DNA is that A, C, G, and T. And so there's the sugar, and then the phosphates, little phosphorus with some oxygen around it. That's the backbone. That's a base pair. That's a base pair. That's a base pair. You got about 3.2 billion in your genome. So we always start with a start codon, the AUG, methionine, whatever, AUG, that's a start codon. And there's multiple stop codons that end it for us. UAG, that's one of them. Could be UAA, could be UGA. But then in the intermediates, we're just coding for different amino acids. So if we were to look at, on this one, to get to methionine, to get to a start codon, what do you have to do? It's AUG, right? If you want those stop codons, UGA and UAG and UAA. And then you can go around to the rest of the amino acids and figure out how to get those ones. So whatever codon is there, whatever nucleotide triplet, that's what determines which amino acid is present. And if you're the Olsen twins, you have the same genome. If you're anybody else, there's a different genome from you and whoever is closest genetically to you. There's a completely different genome. Now, those genomes are made up of alleles, right? Different variants, different versions of genes. So this house is a tiny bit different from that house, a tiny bit different from that house. Like, I guess they all have vans. That one's like a Subaru or something. But the flowers are different. Maybe the shutters are different. This one has an antenna, that one doesn't. So there's very small differences from house to house. There's a single architectural plan, right? There's a single gene, but then there's little modifications, little differences. So those would be alleles, different versions of the same gene. And you can think of it as ice cream cones, right? What flavor of ice cream cone? What allele? It's an ice cream cone. They're the same thing, but they're a little bit different, right? They taste a little bit different. Are the calories pretty much the same? Sure. Is it going to induce diabetes with the same rapidity? Sure. Does the dentist have something to say about each of these equally? Sure. But there's a little difference between these in how you taste them. And so that would be your alleles, right? Different versions of the same thing. Different variants, different versions of the same thing. So blood type. Do you guys know your blood type? Does anybody in here, you know your blood type?
Yeah, A positive.
A positive. I'm O negative, which means my blood is like naked. It has like nothing on it. You guys haven't done like, I guess we don't do that in 129. You don't do it like human phase? Like blood typing, anything like that? So, blood type, there's A, B, and O, right? There are different alleles. And we have different alleles here. And you have two of them, right? If you're AA, you're an A. If you're BB, your blood type is B. If you're AB, your blood type is AB. If you are AO, your blood type is A. If you are BO, your blood type is B. If you're OO, your blood type is O. But there are different alleles here. And depending on where the amino acids, right, so this is amino acid position and residue. So number 156, number 176, like arginine gets replaced by glycine. Methionine gets replaced by arginine. Valine by methionine. Depending on where you are, you're gonna swap some of these out. There's a lot of these different substitutions that can exist there. But we really have 4 different phenotypes that are going to develop. Antigens, they can be carbs, they can be lipids. In this case, they're proteins. They're little proteins. And they're just proteins on red blood cells. The little proteins on your red blood cells. And if you're A blood type, or you have A antigens on your red blood cells. If you're B, you have B antigens on your blood. These little tiny proteins. If you're AB, you have A and B antigens. If you're O, that's no, you don't have antigens there. And then there's the positive and negative stuff, the RH factor, whether you have it or you don't. So I'm O negative, so I have naked red blood cells. Harder to give me a blood transfusion. If you are A, you can receive blood from A or O. If you're B, you can receive it from B or O. If you're AB, you can receive it from A or B. I mean, AB, you have those antigens. Your immune system is not friendly to invasive antigens. But these are the 4 basic phenotypes, right? And this is just the same thing with these different alleles. We get different phenotypes. And those phenotypes matter. It's a little thing with blood type, but go try to get a transfusion with the wrong blood. These little phenotypes matter. You know, again, there's over 70 ABO alleles, but there are only so many phenotypic, manifestations of them. So, how our genes change, how the genome is different, how a gene is different is a nonsense SNP. Remember, that's pencils down. A premature stop codon shows up. A missense SNP, that's the recipe calls for cumin, and you dump a bunch of cinnamon in it, and then everyone else knows it tastes weird, and you're feeling insecure because you don't know what you did to it, and it's horrible. So that's a missense SNP. You put in the wrong ingredient somewhere along the way. If it's a protein, it folds wrong, or it folds differently. And then a frame shift mutation, remember you, in this case, right here, there's a C and the G, these two. And if you pull those out, everything else shifts to the left. So everything beginning at codon 5, what used to be arginine is now glutamate, everything after that changes. Every single amino acid is different. And you may get a premature stop codon showing up that way. It just depends what base pairs are sliding, right? It depends which ones slide. Phenylketonuria, what we're warned about on the back of an equal packet. Same thing, you can't drink milk, you basically can't eat protein. Phenylalanine hydroxylase, remember that's the enzyme that converts phenylalanine to tyrosine. Critical step in the metabolism of phenylalanine. So anything with phenylalanine in it, if you have an allele that causes that enzyme to fold incorrectly, that causes it to fold in a way that cannot carry out that enzymatic conversion of phenylalanine to tyrosine, phenylalanine builds up. Amino acids get to the brain, they cross the blood-brain barrier in a very specific way. In this case, aboard large neutral amino acid transporters, and they're competing. And if you have a huge saturation of one of them, you block transport of the others. You oversaturate those transporters. And so the competing amino acids, the other amino acids that are necessary in your brain can't get there, which is how drinking a glass of milk causes brain damage if you have phenylketonuria, or eating a steak causes brain damage. Sweetening stuff with equal causes brain damage because you can't get the amino acids in there because of one substitution. Position 408, a C becomes a T. There are some other ones, this is the most common. A C becomes a T. So arginine is replaced by tryptophan. Well, actually, we'll talk about this later from the perspective of getting tryptophan, because tryptophan boards the brain through those large neutral amino acid transporters too. And leptin, there are a lot of alleles for leptin, and rodents have them, people have them. If you have a truncated leptin or a dysfunctional, misfolded leptin, you just can't get enough to eat. The appetite never leaves you. You're just ravenous. Even when you look like this, it's like, I don't even know where to put the food. Like my my like cheeks are so fat, I can't even get food in my mouth anymore. And you're still trying to get it in there, right? This appetite just doesn't leave you. And so there's a bunch of different versions of leptin gene mutations, but they all end with excess adiposity and a bottomless appetite. The Belgian blue cows, right? The myostatin knockout, the Belgian blue cows. I mean, that is a set of glutes, right? I mean, this is just like a veiny beast. And these things, they don't have functional myostatin. Myostatin is a myokine, and the gene that makes it, if that gets mutated, premature stop codon shows up, then you don't have that anti-anabolic effect of myostatin. Myostatin is super anti-anabolic. All right, moving into lecture 23, my short one. Congenital insensitivity to pain and anhidrosis, meaning you don't sweat. What you lose is A delta and C nerves. And you can lose these in a couple of different ways, but you don't get, and then your sweat glands are no longer innervated. The A delta, that's that sharp, fast, first responder onset pain. And the C fibers are more dull, achy, lingering pain. But both of these things go away from a couple of different possibilities, both of them genetic mutations. This right here is classic CEPA, which is mutations of TRAC-A, tropomyosin receptor kinase A. That's the receptor for nerve growth factor. You can also have a mutation in nerve growth factor itself. And so if you don't get nerve growth factor, or you don't have TRAC-A, or they're not functional, you don't get that signaling cascade. You don't get that nerve growth factor signaling cascade for the health and survival of your nerves. Same thing, right? This is just CEPA is the absence of A delta and C fibers. You still have A alpha and A beta. You have those, but you don't have C and A delta. Okay, so Travis's lecture, I pulled a few things from it. And beginning with row A, which if you Google row A, you get Real Housewives of Atlanta. So row A, I don't think that's what he was talking about. It's a GTPase protein. We've encountered these things before. Ross homolog, family member A. What else begins with Ross homolog that we've talked about? This is in the Ross superfamily. So you need a GTP bound. So I think this will probably give you some clues. You need a GTP bound to row A for it to be active and do its cell signaling. What else do you need a GTP bound to for it to be active? Red. Ross homolog enriched in brain, right? It's in the Ross superfamily. RAG, not really. It's sort of a cousin. RAG is more of a cousin. But this is in the same family as REG. And so active row A is GTP bound, and it activates ROCK, row A kinase. What does a kinase do?
Phosphorylate.
Phosphorylate stuff. It's going to phosphorylate a bunch of stuff. So there's row A with GTP bound. There's ROCK, and you're going to recognize some of this stuff. Now this is in the heart, PI3K, MNAPK, calcium calmodulin. You recognize some of this stuff in the signaling. Some of this row A ROCK stuff. Now this is from Travis's slides. I did a screenshot of Travis's slides. And what forms these myelin proteins, you're going to get damage of some sort, right? It's it's an injury. It is autoimmune disease. It's something, right? You get these myelin proteins, and we're going to get this complex form. P75, LINGO, NGR. You get this complex form, and that's okay if that complex, if you can get it inside, you activate row A. So there's two things need to happen. You need to form this complex of proteins, just like mTOR is a complex, right? There's a bunch of stuff in mTOR, it's a whole complex. You form this complex, but you also have to get it inside. If you just form the complex and you can't transport it No crisis. You're not activating Rho A. Rho A is inhibiting both myelin and the growth of the neuron itself. So to get it inside is that LRP1 receptor, that low density lipoprotein receptor, lipoprotein 1. And this is the pathway that Travis is working with Novaron. It's a huge protein complex with diverse signaling, but one of its functions is to get that complex inside so it can activate Rho A. So he went through these questions. This is just another copied and pasted another screenshot of his slides, and he just answered yes to all of them. Right? So if you listen to the lecture, he's like, does blah, blah, blah? Yes. Does, like, have you ever seen Bob the Builder? Familiar with, can we build it? Yes, we can. It's like the exact same thing that Obama said when he was running. Can we, whatever? Yes, we can. That's sort of the idea. We have these 6 questions, and he goes through it a little bit and says yes. But internalizing, right, this structure, getting it inside so it can activate Rho A. If Rho A is active, you are abolishing neural regeneration. If Rho A is active. And so that is the pathway that he found of this LRP1 is the pathway that can get these things internalized to activate Rho A. Rho A GTP, so it can do its signaling. And in this case, its signaling is let's abolish neural regeneration. Are we okay with the Travis Stiles lectures? Okay, so let's move on to telomeres. Telomeres are, who has shoelaces? Okay. Amanda has shoelaces on, and at the end of shoelaces, and Cameron has shoelaces, lots of you have shoelaces. At the very end of them, there's an aglet. It's a little plastic cap. Sometimes they're metal, but it's a little plastic cap at the end of your shoelaces. That's essentially the genetic equivalent of what telomeres are. So this is genome.gov. It's the end of a chromosome, a telomere. The end of a chromosome, they're made of repetitive sequences of non-coding DNA. So these telomeres do not code for proteins that protect the chromosome from damage, but every single time the cell divides, the telomeres shorten. Every time cells divide, those telomeres shorten, and they get shorter and shorter and shorter until you can no longer divide. So here's the aglet, this little thing. That's the telomere right there. And let's say every time you tie your shoes, let's say you don't tie your shoes that often, but every time you do, the aglet shortens just a little bit and shortens a little bit and shortens a little bit. Eventually, you're going to run out of aglet, right? You're going to run out of that cap, and the shoelaces fray. Genetically, the telomeres serve a very similar function. They get shortened every time the cell divides. So right here, at the ends of a chromosome, you're seeing those telomeres. And they keep shortening and shortening and shortening until you can no longer divide that cell. Or apoptosis happens, right? Either you can't divide or apoptosis. So telomeres are conserved tandem repeats at the chromosomal ends that differ in length in diverse species. Rats have really long telomeres. Invertebrates, like you have a spine, chromosomal ends consist of TTA, GGG repeats. So it's just over and over and over. TTA, GGG, TTA, GGG. Human telomeres typically range between 10 to 15,000 base pairs is the general length of telomeres. Now from Nature, one of the top journals in the world, human telomeres, for example, range in size from 2 to 50. So 10 to 15 is usual, but they can range from 2 to 50 kilobases, 50,000 base pairs, and consist of approximately 300 to 8,000 precise repeats of the sequence CCC TAA, right? Just if you flip it over, TTA GGG. A, T, C, 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 right? So TTA, GGG, that's usually how it's written. TTA, GGG, TTA, GGG, TTA, GGG, just over and over and over and over. It doesn't code for any proteins. It's just there as an aglet. Or like an odometer. Think of it as like an odometer. This tells you, it's a marker for age. Replicative senescence, right? This how many replications have there been? Just keep dinging off on that odometer over and over. So this one, pretty much everything in this passage is good. So I'm just going to read line by line. Normal human cells in the culture stop dividing after 40 to 60 passages. You don't just divide forever. You don't just divide forever. First observed by Leonard Hayflick. So it's the Hayflick limit. The Hayflick limit, that's like 1961. It's really old. Didn't understand this, how telomeres doing this and how it works, just that there's a limit, right? So that was in the 60s, that there's a limit. Incomplete replication with a gradual shortening of telomeres acts like as a counting mechanism that eventually leads to replicative senescence. Aging at this cellular level where the cells can no longer divide. On average, a single human telomere contains enough repeats to buttress the effect of telomere erosion in the absence of a maintenance mechanism, with an estimated loss of about 50 to 250 base pairs per mitosis. So the number of base pairs that gets hacked off of that telomere, of that aglet, with every replication. Telomere shortening is mitigated by telomerase. And so this is an enzyme that does the repairing. Now, if you're a lobster, or if you could be like hybridized with a lobster, you would have really effective telomerase. You'd be putting that stuff back together. Lobsters live a really long time, which is why it's so mean to eat them. You know, if you eat an animal that's like, well, it's gonna die in a year anyway. Like, let's just, to put it in perspective, what's the average lifespan of a dog? 14 years, something like that. So if it's like a 10-year-old dog, you've denied the dog 4 years. A lobster, you're eating these lobsters like 2 years old. The thing might have had a few hundred more years of life in it. It's going to die from other reasons, but it has this really potent telomerase. It doesn't have the same version of aging that we do. It doesn't mean it lives like, you know, as long as the sun, but it's a little bit mean to take a couple hundred years off a creature's life because it tastes good with butter. Anyway, we can't just infinitely replicate our cells. They eventually expire. The odometer counts backwards, and like when it hits zero, that car is not running anymore. Now, per se, nobody gets that word right. That just means by itself. You know, what people would say, like, well, it's not a sports car per se, but it really does have some go. That doesn't make any sense. Like per se doesn't mean exactly. Per se means on its own. Acapella, you know, that's the same thing as per se. Aging per se is the major, so they got it right, but most people get it wrong. Aging per se is the major risk factor for most pathological conditions that limit the health span and promote chronic disorders affecting the elderly, including immunosenescence, cardiometabolic disorders, osteoporosis, sarcopenia, just loss of muscle, arthritis, cataracts, neurodegenerative diseases, and most cancers. Aging is our risk factor for pretty much everything. No matter what ails you, no matter what condition you're worried about, aging is a huge risk factor for these things. And so what is it about aging that puts us at so much risk? What is it within aging that bestows all of this risk on us, this stuff we don't want? Now, you can tell this isn't human because there's no A in there, but here's the telomeres in some critter, and telomerase doing its fixing. Telomere ACE, it's an enzyme, an ACE, telomerase, doing its fixing. So here's the chromosome of an adult cell with the telomeres at the end, little caps, right? The telomere is shortening because the cell has been replicating, right? We have cell division and it's shortening the telomeres. At senescence, at aging, right? Now we are, the health span is over. We are now entering into those disease years. We're entering into those years of hardship that precede death. But in a mortal cell, you're going to have telomerase on there fixing it. Cancer does a really good job of immortalizing itself. Cancer cells. So if we were to hybridize with a lobster, maybe like reverse transcriptase some like lobster sauce into us, and we were to have very effective telomerase, it would probably put us at a huge risk of cancer, because this is cancer, right? This is this replication without consequence, replication without an odometer monitoring it. This whole passage is good here as well, where just as a summarizing passage, the telomeres are gene poor, right? We're not coding for anything here, whatever. Repetitions of TTA, GGG in people, right? That's us. TTA, GGG, TTA, GGG, TTA, GGG, over and over and over for this, capping the end of chromosomes. And they help stabilize the chromosomes, preventing degradation, end-to-end fusion, and abnormal recombinations of DNA strands. So they're critical for your DNA, right? For the health of your DNA. With each cell cycle, they shorten 30 to 200 nucleotides, right? Maybe 100 base pairs get wiped out every single time with each cell cycle, every single time they replicate. This process is further accelerated as a result of oxidative stress and inflammation. Now that brings us to behaviors, to nutrition, to exercise, to environmental exposure. If we have oxidative stress, and if we have an exaggerated inflammatory response, we shorten our telomeres more and more and more. Biological aging happens faster and faster and faster. We all know people who age well and age badly. There are people who, they're like 60 years old and they look 32 and they act 32 and they tie their shoes like a 32-year-old. And then there are people who are 32 who act and look 60. And we've all encountered these people. Maybe not that extreme, but when somebody says, "Can you guess how old I am?" And then you look at them, and nobody ever is honest with that question. Because what that question is doing is, "Will you pay me a compliment?" That's all anyone says. But if it's like a picture, and you're thinking, "How old is that person? How old is that person? How old is that person?" We would guess somewhere in the ballpark of accurate, and once in a while we get it way off. Either that person's super old and they're not, or that person's super young and they're not. But they kind of are. Biologically, age really is different. It doesn't matter how many times the Earth circles the sun. Nobody gives a shit about that. It's your aging process is biologically different. The rate of the onset of senescence, the rate of shortening those telomeres is different from person to person. And this matters. This rate matters, right? You don't even really have to read this one. Just telomere shortening rate predicts species lifespan. The title is bold enough. Now, you can't compare one species to the next and say like, well, rats, super long telomeres. Why aren't rats outliving lobsters? You know, one species to the next, but the rate of shortening when you look in an individual species, and all we really care about is people and maybe your pets, and looking at the rate of telomere shortening in us is a massive predictor of lifespan. Now, Elizabeth Blackburn won a Nobel Prize for discovering how telomeres and telomerase work. So this is, it's a book that's written really for the lay person. In stress physiology, we did 5 lectures on sleep health, sleep hygiene and sleep health. And Matthew Walker, he's over at Berkeley, he wrote a book called Why We Sleep. and it's a great science book for lay people. This is the equivalent of Why We Sleep, about telomeres, right? But she's the one she's really the biggest name in this. I actually don't know anything about her. They both, I think, narrate it together. I did Audible version, which is why I say they narrated it together. But in that book, they go through all of these different behaviors and exposures that change the rate of biological aging, of telomere shortening, and potentially telomere lengthening. We do have telomerase. We can't compete with a lobster. We're bad at that part. What we're good at is slowing the rate of aging. Now, caffeine is one of them. You're gonna get sick of Larry Tucker by the end of the lecture. If there's something about food and telomere length, Larry Tucker is just like every time, and he's the guy. He does all this stuff. The N. Haynes data set, huge epidemiological data set. But what he's looking at in this study is caffeine, how much caffeine people have, and the lengths of their telomeres. Now, let's just look at men and women combined. There's 5,800 people. 5th percentile of caffeine, you're having quite a bit of caffeine. 49, 43, right? That's the base pairs, the number of base pairs. 49, 43. Back off on caffeine a little bit. 53, 73. Oh, telomeres got longer. Back off on caffeine a little bit more. 57, 38. Got longer. Back off on caffeine a little more. 61, 88. Okay, we're getting pretty long in our telomeres. Back off of caffeine. 7014. There are incremental doses of caffeine. The more caffeine. Now, caffeine is wonderful. Caffeine has a ton of ergogenic properties to it. Caffeine is, it's helpful for focus and for almost any sport, aerobic, anaerobic. Caffeine is really helpful, but it's shortening telomeres, as the evidence indicates. Now, interestingly, coffee lengthens telomeres. Caffeine shortens telomeres. It's probably other components in caffeine, in coffee. Coffee is very rich in polyphenols. And when you look at a lot of the literature, decaf coffee usually has the exact same health benefits as caffeinated coffee. When you look at the consequences, caffeine is sometimes associated with the consequences and decaf is not. And so this is, when you see these mugs, death before decaf, that's predictive, right? That's literally. Decaf coffee in this sense, in this study, and it was a pretty compelling study, the way they did it, where every single angle that they looked at it, they found the exact same pattern within groups. It was a pretty compelling look at how they did it. Adjusting for a bunch of covariates. So it's regression modeling. Let's control some stuff. And, but the decaf seems to be a prolonger of telomeres, and caffeine itself seems to.
What was the age range?
Yeah, it's 20 something, 28 to 82. It's something like that. Good question. Yeah, it's 20s to 80s.
Did they do any, I guess, diagrams or models on like the different age ranges?
They might have looked at individual age groups, but this was just across the lifespan. And when they were looking at, there might be one here. To this, it comes close, I think, to answering your question. In the present investigation, the inverse relationship between caffeine intake and telomere length, so more caffeine associates with shorter telomeres, was dose response and consistent. After adjusting for the covariates, adults with no caffeine intake had the longest telomeres, and those with the highest caffeine intakes had the shortest. Moreover, the dose response pattern prevailed across all of the coffee subgroups, coffee drinkers only, those reporting no coffee intake, and when all participants were studied together. So they divided this study a ton of different ways and reinforced the analysis, and every single time it came out. The more caffeine you have, the shorter your telomeres. Now, I'm not saying ward yourself off caffeine. Definitely don't ward yourself off coffee. Coffee emerges in so many studies, in cancer studies too, as like this incredibly anti-carcinogenic, depending on the type of cancer, colon cancer, stuff like that. And this ergogenic aid in a number of disciplines. And caffeine is really effective. Coffee is really helpful. But one potential complication of caffeine is shortened telomeres. Cigarettes, everybody could expect this one coming, right? Smoking, and you're going to shorten your telomeres. And you can look at this in tons of different ways. But this one is current smokers compared to non-smokers. And here's no difference right here. Each one of these is a different study, a study that has evaluated this, a ton of different studies. Each one of these has 214 smokers compared to 421 non-smokers, 525 smokers compared to 2,000 non-smokers. So each one of these is an entire study. Some of them are pretty big, 500 smokers, 2,500 non-smokers. But in all of these studies, this is the average effect, where a smoker, a current smoker compared to a non-current smoker, meaning maybe they used to smoke, but they don't smoke now, right? They stopped smoking a little while ago, and so they're technically non-smokers now, or they never smoked, however you want to look at it. But this can include previous smokers. So current smokers as compared to non-current smokers. Telomeres are shortened. So that's the mean effect in a meta-analysis, the mean effect of shortening of telomeres with cigarettes. And this one, same study, right? Same meta-analysis, a bunch of different studies they're comparing in this one because it's ever smoked compared to never smoked. So not necessarily current smokers, but have you ever smoked? Yes or no? Have you ever smoked? Yes or no? The yeses and the nos. And there's the same pattern. Smokers and previous smokers have shortened telomeres. Now, I smoked a while ago and I quit. Doesn't have that big of a difference. Doesn't seem to have that big of a difference. So quitting smoking is a way of helping to keep telomere length intact. Now, when you're drinking water, they always say BPA free. Now they do. They didn't used to do that. Before they didn't used to say anything. They just said like Aquafina, drink now. But now there's always some little like BPA free on these water bottles. And people are looking at the risk of cancer. People are looking at all these possible risks. A remarkable number of in vitro, like in glass, right? Not in a body, and in vivo, meaning in the body. Studies on low-dose BPA exposure have identified a potential risk of BPA for adverse health outcomes. Tons of different adverse health outcomes. What might they be related to? Well, we see a shortening of telomeres with BPA exposure. And BPA exposure, you breathe in dust. It's not just water bottles, receipts, and you breathe in the dust. It's like everywhere, this stuff. And BPA isn't the only thing. This is just an example of an environmental exposure that we find ourselves overexposed to for a while, and then kind of political positions. Oh, we have to make them BPA free for the water bottles. Okay, that's one step. Pesticides, this is another. Now, most of the work on pesticides is not in people who go to S-Mart and decide whether to buy organic or inorganic. It's just, we're accepting the word organic as like, pesticide, as opposed to like hydrocarbon bonds. There's like no such thing as, organic water. And there's no such thing as an inorganic apple. If you like organic chemistry, right, it has carbon in it. But organic as a grocery term, meaning, do they use pesticides, stuff like that. There's a lot of literature outside of telomeres that shows things like upregulating aromatase enzymes. That's the thing that converts testosterone to estradiol. And there's a lot of research on the effects of pesticides. This is pesticide like workers, like the farmers who are exposed. And you do see in almost all of the pesticides evidence of shortening of telomeres. Not in all of them. for the RTL, that's a relative telomere length shortening. RTL, that's a relative length of telomeres shortening for a lifetime, whatever. No telomere lengthening was observed for any pesticide, but most pesticides associate with telomere shortening. These doses would be a little bit higher than what you'd be getting in the grocery store, though. Basal cortisol, stress levels, right? This article, it does have a nice little summary, just a reminder of this stuff. Nobody needs this reminder, but telomeres are repeated nucleotide sequences, TTAGG, that act as protective caps at the ends of chromosomes. Telomeres shorten with each cell division, and when telomere length reaches a critical threshold, cells stop dividing or die. So just a little reminder. But the HPA axis, the hypothalamic pituitary adrenal axis, if we're going to have a bunch of cortisol release, stress hormone, emotional stress, physical stress, psychological, whatever stresses you out, we're going to have an acute response with the sympathoadrenal medullary system, epinephrine, and the HPA axis to get cortisol. So this acute stress response. Now, people who have elevated basal cortisol levels, there's not that much of an association with telomere length. It's an insignificant, meaning P doesn't reach 0.05, or it's the effect is modest and inconsistent. But the way that people respond to acute stressors, if a stressor brings an exaggerated reactive response, right? If a stressor, induces this elevated kind of peak of cortisol, of reactivity, now we're seeing shortened telomeres. So this is like, go find your Zen space. For cortisol reactivity and telomere length, we're seeing shortened telomeres in most studies. Again, these are all different studies with different samples, and this is the mean of all studies right here. So where might we find these stressors? Social relationships, right? As people age, social health becomes really important. Have you ever had a grandpa or grandma or something who like lived alone for a while and they got super weird, and then they like found somebody again and they unweirded themselves and they became like normal again? Does anybody have a family history where you have like an elderly person who had isolation and they got weird for a bit, and then isolation stopped and they found themselves in a healthy societal environment, and like everything was healthy and all their ailments started to heal? This is sort of a common experience for people. If you have a healthy system of social support, we're seeing elongated telomeres, longer length of telomeres in those people. Bad relationships. If you find yourself in bad relationships, you're going to see shortened telomeres. Now, healthy relationships, this article goes on to talk about, like, did you have sex in the last week? Your telomeres are longer. Characterizing what a healthy relationship looks like in a romantic context. But really what's important is, are relationships in your life healthy? Your telomeres applaud you. Are relationships in your life unhealthy? Telomeres shorten, which means you age faster, which means you probably die younger. And those last years of life are filled with disease, probably. This stuff is important, right? It's important to maintain this stuff. And so even just looking at people who are married or were never married compared to people who had a rocky marriage and got divorced, telomere length differences. Now, nutrition. Remember earlier I said oxidative stress and inflammation. Oxidative stress and inflammation, systemic inflammation and oxidative stress are huge causes, are huge culprits of an advancing truncation, this truncating of your telomeres. We can see something similar in nutrition, right? Nutrition, I mean, we all know antioxidants, right? Go eat your vitamin C and your vitamin E and your lycopene and your whatevers. All these antioxidants. We're warding off oxidative stress. Okay. Other foods we get an inflammatory response from. Now, it's not all oxidative stress and inflammation. This is one. I said you'd get sick of Larry Tucker. So here's getting back into some of these foods. Larry Tucker saying the milk fat is associated with shortened telomeres. And part of what he's saying in this article is anabolic signaling. Right? I mean, he talks about mTOR, you know, in this article, PKB, mTOR stuff. And it's, there's a bunch of branched chain amino acids in milk and what. But saturated fats and dairy saturated fat and there may be an inflammatory response or maybe additional mTOR signaling. But what he found was an increased biological rate of aging, telomere shortening, compared with those who drink off-fat milk, where about 145 telomere base pairs representing years of increased biological aging. So just the number of telomeres changing with milk. Tucker again, dietary fiber and telomere length. And really using the same sample over and over and looking at individual components and holding constant a bunch of potential confounders. So here, what he was holding constant was smoking, BMI, alcohol use. Excesses of alcohol use are associated with shortened telomeres, but not, and physical activity, as well as other covariates. But those are the main covariates. So each 10-gram increase in daily fiber intake associated with 67 base pairs longer of telomeres, a biologic aging difference of about 4.3 years. So fiber, right? So we're seeing dairy fat being associating with shortened telomeres. Now, association is an important term here because it wasn't a randomized controlled trial where everyone was doing the same thing, randomized into groups, and you're the milk fat group, and let's see what happens to your telomeres. This is epidemiology. And so there's room for error in here. From the perspective of the type of people who eat low fiber, the type of people who eat a lot of dairy fat, the type of people who do those things, maybe the type of people who don't have good sleep hygiene. And maybe sleep is the real culprit. See, a lot of these, the type of people who is a limitation to epidemiology. So that's the dietary fiber one. Nuts and seeds, we got Tucker again. And nuts and seeds, people whose diets contain a higher percentage of the calories coming from nuts and seeds have longer telomeres. So if it's 5%, for people who, if they consume 5% of their total energy from nuts and seeds, they're looking about a year and a half of reduced cell aging compared to the no nuts and seeds folks. So having nuts and seeds, having high fiber, low dairy fat, any foods that are going to be very inflammatory, try to avoid that stuff. But telomeres themselves, it's not the only determinant of aging. Telomeres, while they're interesting and while, the Nobel Prize was, I want to say it was like 2009 or something. It's not that long ago, figuring out how these things work. And there's room for pharmacological interventions, right? There's room for exercise and nutritional interventions. But there's a lot of components of aging, and telomere, this is one of them. And so not to overemphasize this point, not to get carried away with it. In this article, they talk about a bunch of different characteristics of aging. You don't need to know any of these. I'm not going to ask you about any of these characteristics of aging. This is just for context. This is just to say that telomeres aren't everything. They do talk about telomeres, telomere attrition. That's one of the components. Genomic instability beyond the telomeres. Right, DNA damage accumulating, and that is going to change with age. Epigenetic alterations, histone modifications, methylation. We're going to control the genome. Epi meaning upon, epigenetics. We're controlling it through this sort of physiological states that are not the DNA itself. And so epigenetic alterations can happen. Loss of proteostasis, right? Protein, we're looking at the metabolism of our proteins can go awry. Nutrient sensing can get messed up. This could be insulin and is mTOR on or off, signaling for nutrients. Mitochondrial dysfunction. As we go through life, we see more and more dysfunction of the mitochondria, cellular senescence itself, stem cell exhaustion, altered intracellular communication. So a lot of the cell inflammation can really mess this up. So there's a lot of components of aging. There are a lot of explanations of aging. There are a lot of areas in which we exhibit aging. Telomeres, that's one of them. And it seems to be an important one. And it seems to be one that we can control with our behaviors. So at the start of today, I said I would show you a very simple summary of what to do with older adults. So from the same book, the 11th edition, ACSM, testing and prescription guidelines, page 177, they're talking about older adults. And older adults, remember, not everybody ages at the same rate. We've just been talking about telomeres, right? The rate of telomere shortening is a major determinant of lifespan and our biological rate of aging. The number of years the number of birthdays you had doesn't really matter that much. What matters is physiological health. So there's a couple of different ways of looking at what an older adult actually is. Greater than or equal to age 65. You're an older adult, I don't care who you are, you're classified as an older adult. Ages 50 to 64 with clinically significant conditions. So if you're older than 50 and your biological age is much older than 50, but you're not 65 according to the number of birthdays you had, you're between those years, but your biological age, right, there's some clinical significance to your conditions. That's also considered an older adult. You can be a, quote, older adult at age 50. Now, there's other stuff you're going to want to do. What you have to do if you're working with anybody is that chapter 2 stuff. Stratify the people into their risk classes. Figure out how many risk factors they have. Estimate likelihood of having an adverse event. And decide on an intensity, on an exercise prescription that is appropriate to the person's current standing, their physical capacities. But also with older adults, you need to figure out their balance, right? Older adults fall a lot, and a fractured hip, that really sends us spiraling. You can fracture a hip and be like totally normal. If like an 80-year-old fractures a hip, and they were normal before, no dementia, their cognitive functioning is just as it was when they were 25. They fracture their hip, 2 weeks later, they can't even like remember their birthday. This can be really bad. So figuring out mobility and balance. There's a lot of tests that you can do. And then this will be our last slide. This is what ACSM recommends for older adults. And for aerobic resistance and flexibility, resistance at least 2 days a week. Don't care who you are. Resistance training is for you. When we see older adults experiencing atrophy, those type 2 fibers go because they don't use them. Right? They just become type 1 fiber creatures. It's the use it or lose it, right? The type 2 fibers are just gone. And so trying to preserve some strength, some physical capacity, can they carry groceries up a flight of stairs, or strength training, resistance training has really been emphasized in the last decade or so with older adults. Also having some flexibility so they can continue to get dressed. So it's not just like, oh, I can't, so they have some mobility. And then the aerobic part, at least 5 days a week of moderate, vigorous, at least 3 days a week if you can get that, with the time and types of things. What questions do we have? Okay, yeah.
Just to be clear, so with the telomere lengthening, is that equivalent to an increase in telomere activity for repair?
The telomerase, the enzymatic repair. Yeah, so it would be. We have very limited telomerase, and the evidence of lengthening is weak at this point. Sometimes what we're seeing is epidemiological studies, but there are hints in the research of telomerase activity in people having a lengthening, but it's not like we're going to reverse aging, from that perspective. It's not like, well, last year I was 41, but I'm going to be 40 at my next birthday. It's not quite to that extent.
So then is it more so saying that there's always going to be that kind of shortening, but it's not as much? Like
That is the most practical way of looking at it, where as we go through life, it shortens. Our telomeres shorten. They just get clipped and clipped and clipped and clipped. And is there a possibility to lengthen them? Sure, but really what we're trying to do is mitigate the damage, mitigate the shortening with healthy behaviors. And we'll talk more about exercise as we move into cognitive health and movement and stuff.
What is like for smoking and how it shortens telomeres?
What's the mechanism?
No, like when smoking promotes cancer, and you have undying telomere cancer.
Oh, I see what you're saying. Yeah, it's a two sort of different contexts. When you look at immortal cell lines, they just, like those telomeres don't shorten. And so if you're in a cancerous cell, that's different from being in whatever other tissue, cancer-free tissue. If you're in a cancerous cell, you're looking at potentially this immortalized, cell line. So if you smoke and smoke and smoke and get lung cancer, and then that cancer can repair itself, that's sort of a different situation. Because the rest of your telomeres aren't just like, oh, us too. It's like monkey see, monkey do. I see what you're doing. I'm going to do it too, and now I'm living forever too. So it doesn't quite have that association. Yeah, two sort of different environmental worlds, biological worlds. All right. I will see everyone on Wednesday.